Hello guys, so today we're going to talk about muscle contraction. Let's start with the macro anatomy. So this is what I have for you here. This is a skeletal muscle, right? So if you do a cross section of a skeletal muscle, what would you see inside as a slice, right? You would see here is the skeletal muscle surrounded by a membrane called epimysium. Inside you have these smaller structures that are called fascicles. Each fascicle is surrounded by a perimysium, which is this membrane right here. And then inside the fascicle you have individual muscle fibers, and when I say muscle fiber I mean muscle cell. And each cell is surrounded by a membrane too that is called endomysium. Okay? If you take, for example, let's say here we have 3 individual muscle fibers or 3 individual muscle cells. One motor neuron that comes from the central nervous system is going to go all the way through the peripheral nerves and innervate, for example, these 3 fibers. So we call the group of one motor neuron and the muscle fibers that it innervates a motor unit. Okay, so this is a concept that we need to know. One neuron that is innervating 3 fibers all together are going to be called a motor unit. It could be more than 3 fibers. Okay, now we're going to try to understand what is happening here for contraction to happen. And this is when we move on to the microanatomy. So this is what we have. Muscle fiber is here. I'm looking at a little tiny portion of it. So we have here this line here that represents the plasma membrane. In a muscle fiber, you call it sarcolemma. So sarcolemma is the plasma membrane. And this is an axon terminal from the motor neuron. So here you're going to have little vesicles that release in the synaptic cleft, which is this space, acetylcholine. So this little molecule here is called acetylcholine. Acetylcholine binds to this receptor. So this is the acetylcholine receptor. And acetylcholine receptor is coupled with the sodiums for channel. So remember how the plasma membrane is always polarized, so it's positive outside, negative inside. So we want this membrane to depolarize to allow for muscle contraction. How does the membrane depolarize? Sodium goes in, so you have an influx of sodium. When sodium goes in, the membrane becomes more positive inside than on the outside. And this is going to activate other channels that you have along the sarcolemma this is chemically gated, right? Acetylcholine opens this gate and sodium goes in. Here, these ones are voltage gated. So because of this difference in potential here, these gates open and then more sodium goes in. Oops. Okay, more sodium is going in. So this depolarization continues all the way to here. So negative outside, positive inside, right? And when this is happening, it also activates voltage-gated channels that you have in the membrane of the sarcoplasmic reticulum. The sarcoplasmic reticulum is a cisterna that is full of calcium. And then as these voltage-gated calcium channels open, calcium goes out. Okay? Okay. So what I told you so far is step 1. So step 1 is depolarization and calcium release. Now we're going to talk about step 2. Step 2 is the cross bridge. Okay, so here we're talking about cross bridge. Which are the components for the cross bridge? You have actin or thin filaments, which are the green ones here, and myosin or thick filaments, which are the brown ones. Okay? You also have this orange filament here that is called troponin. Troponin is attached to actin in the absence of calcium. Once calcium is playing his role, troponin detaches from actin. So then troponin is here now, detached from my actin. Okay? What happens when that event takes place? The sites on actin where myosin can bind get exposed, and then the myosin heads can bind to the actin. And by binding to it, they slide. So this is the sliding mechanism. And I'm going to call this number 3. You see how here myosin was not aligned with actin, and here they are aligned? So the sliding mechanism leads to a 4th event that is called sarcomere shortening. What is that? You are shortening the size of this whole. So let's see if we can measure here. If I had a ruler, I would measure this, right? And then you compare to this. You see how it's shortened? Okay, so that's the shortening. And just so you know, a sarcomere would be this, and another one here, and another myosin here, doing the same thing. So I'm just showing you here half of a sarcomere. Okay? How do you undo this mechanism? You need ATP. ATP is going to detach the myosin head from the actin sites. Okay? So when that detaches, you break down ATP. You need energy for detachment. And as they detach, they continue to be able to reattach in other sites. So this continues to move kind of like a leg walking on a path, right? A foot walking on a path, for example. So that phenomenon is dependent on actin. And how does this end? So the end of it is when on the sarcoplasma, or sarcolemma, sorry, you have other channels. Let me make them a different color here. Let's make them green. So these channels now are for potassium, and they're also voltage gated. So the voltage that is created by this whole mechanism allows potassium to go out. Okay? And as potassium is going out, you repolarize the membrane. So instead of depolarized, now the membrane is going to be all repolarized. And when this happens, the muscle relaxes. Why? What is going to happen here when muscle relaxes? There's no more calcium. Calcium returns to the sarcoplasmic reticulum, and troponin goes back to protecting actin, so myosin cannot bind to it. I hope this mechanism is clear. Let me know if you have any questions.